This is Dr. Ibrahim. He is an associate professor of cardiology and a research scientist at the Smith Heart Institute at Cedars-Sinai Medical Center. His research focuses on the translational applications of extracellular vesicles and their RNA constituents, investigating how their cargo of small RNAs, lipids, and proteins affect healing and regeneration across diseases. So I usually think of TREX1 with RVCL, but I'm interested to hear it in a new context. So today he will talk about TREX1 upregulation in macrophages as a strategy for promoting transplant tolerance. Welcome.
Well, thank you so much. And I'd like to thank all the organizers for this wonderful event. This is the first transplant workshop I attend, and many thanks to Peter for this wonderful invitation, for Yvonne for helping me coordinate from a distance. Let me share my screen real quick. Okay. Can everyone see my screen?
Almost.
Yes.
Okay. Let me see if I can get this almost to a yes.
I think it's on our side. So,
Oh, I see. Okay.
yeah.
I'm sorry, Ibrahim, can you please share again?
Sure thing.
You had it up, but you took it down.
How's that?
Yep, just hold there. Thanks.
Okay, perfect. Well, let me know if the slides don't proceed. But anyway, thanks again. The title of this talk is TREX1 upregulation in macrophages as a strategy for promoting transplant tolerance. Now, full disclosure, I don't work in transplant biology. I'm more of an RNA biologist, and I focus primarily on RNAs from extracellular vesicles, which have gained a lot of traction in the regenerative medicine field. But today I'll talk about how some of these RNAs that we're discovering may have relevance to other disease contexts beside regenerative medicine.
And we can see your slides now.
Okay, perfect. Yeah, is it progressing? Can you see it?
Yes.
Okay. So the whole message of today's talk, really, that I'd love to share with you is that extracellular vesicles from therapeutic cells contain a whole catalog of small non-coding RNAs, the function of which are unknown. As you all know, 98% of the genome is non-coding RNA, and more than 90% of it is of unknown function. I mean, we understand microRNAs, siRNAs, long non-coding RNAs, maybe, but there's a whole host of small RNAs that have untold function and thus untold potential for a therapeutic application. What you see here, if you can see my mouse, are the RNA distributions in the EV populations from this therapeutic cell type. These are cardiosphere-derived cells. I won't go too much into them, but they have cleared phase 3 clinical trials for Duchenne muscular dystrophy. And then on the left is the RNA distribution inside the EVs of a therapeutically inert cell type. It's a fibroblast. You can appreciate the differences in the RNA distributions and what these EVs contain. And what I'll talk about today is how we discovered a small RNA from these therapeutic cells and then transitioned them into sort of a drug product. This RNA is called NT4. It came as two variants, a shorter one and a much longer one that we call EVYF1. And both of these derive from the human YRNA4 gene. Now, YRNAs, their function is relatively unknown. People may think they might be involved in cancer processes, but they also have they've been implicated in autoimmune disease, and they've been shown to be important for stabilizing DNA replication. But be that as it may, this is a fragment that was discovered and it derives from this human YRNA4 gene. And so when we found out that this RNA fragment is in and of itself therapeutic, we decided to develop a drug out of it. And the way that we did that was we used this IL-10 reporter assay to hone this drug through 3 stages of development. First, through primary mutation of the sequence, then the inclusion of locked nucleic acids, and then the inclusion of other chemical modifications. And then so the final drug candidate that we developed is this thing called TY1, which has a GDC mutation on the 5' end, and then locked nucleic acids 3 alternating on both the 5' and the 3' end. And what you see below here is just the scramble sequence for control purposes. What you see here is resistance to degradation to RNase-R, which is a testament to the effectiveness of the inclusion of locked nucleic acids. Now, in trying to figure out what the mechanism of this TY1 drug is, that's, again, bio-inspired by a naturally occurring RNA in the EVs of these therapeutic cells, we found that the DNA damage response came up the highest. And at that point, we had never really understood DNA damage. We never thought about it, but the data sort of led us there. And if you look at the bottom of this heat map, you find that this pathway came up very strongly associated, which is TREX1 and then the C-gasting pathway. Now, just a very quick primer. TREX1 is this 3' exonuclease that's expressed that's present both in the nucleus and in the cytosol. In the nucleus, it's part of the DNA repair complex. In the cytosol, it degrades leaking cytoplasmic and mitochondrial DNA as a way to reduce cell stress. The C-gasting pathway is a very primordial pathway designed to activate inflammatory genes when it detects cytoplasmic DNA. And this probably comes back from, the war between bacteria and bacteriophages, and so it's a very ancient pathway. We found that in a rat model of myocardial infarction, intravenous injection of this TY1, this drug, reduces DNA damage as the sequencing had predicted. And we also found upregulation of TREX1. And when we tried to find out what the mechanism was, we did an RNA-RNA pull down using 2 methods. And what we found was that when we take the consensus RNA targets between both methods and then cross-reference it with the original macrophage TY1 sequencing, we find that TREX1 pops up in all 3 of these data sets. And then when we do 5' and 3' luciferase binding assays, we find that TY1 binds the 5' UTR of TREX1. Another interesting feature that we found, and one probably inspired by our work with CDC EVs, was that TY1 in vivo depends on the presence of macrophages. And we know that because when we take animals and we inject them with claudinate, these are these toxic liposomes that are taken up by phagocytes primarily, and it kills them. So we deplete the animal of circulating monocytes and macrophages, and then we induce MI, we induce myocardial infarction, and then give TY1 20 minutes after reperfusion. We find that the animals that don't have intact macrophages do not have a reduction in the infarct as the ones that have intact macrophages do. So this shows that macrophages are necessary for TY1 mechanism of action. At the same time, if we, instead of injecting the drug itself, if we instead incubate bone marrow-derived macrophages with either TY1 or upregulate TREX1 directly, we find that the full benefits are recapitulated. However, if we incubate the macrophages with TY1 and abrogate its ability to upregulate TREX1, that effect is lost. So this is sort of showing that macrophages are both necessary and sufficient target cells for TY1 mechanism of action. The other interesting thing that we found, and this was found in response to a reviewer comment, was, well, the reviewer said fine, but how do you connect macrophages upregulating TREX1 to the cardiac repair that you see in cardiac tissue? And what we found was that macrophages change their extracellular vesicle secretome in response to this TY1. And first we find this by when we adoptively transfer macrophages that were given TY1, but are also given GW4869. This is an inhibitor of nitrosphingomyelinase, and so these macrophages are unable to secrete EVs. We find that both the infarct size and the circulating disease marker cardiac troponin are both unchanged. Furthermore, we find that if you gave TY1 directly to injured myocytes in vitro, that the therapeutic effect was absent. However, if you incubate them with macrophages that were transfected with TY1 or macrophages, EVs from macrophages that upregulate TREX1, then you see a protective effect. And indeed, when you transplant those EVs alone, you find that the full benefits are recapitulated, which again points to this, connects the circuit of TR1 gets upregulated, gets uptaken by macrophages. Macrophages secrete EVs. The EVs do the tissue repair. And so this is sort of the overview of the mechanism that I mentioned earlier. So in times of stress, whether it's sterile or infectious injury, DNA damage goes up and C-gas sting also goes up. But with TY1 treatment, TREX1 goes up, DNA damage goes down, immunomodulation happens, and then tissue repair happens probably through the secretion of an altered, more tissue reparative macrophage EV subset. Now I want to shift gears here really quickly and talk about sort of what's more relevant to this exciting group. And before I go into this, I want to thank a very talented surgical resident. Her name is Tiffany Lim. She works under the mentorship of Dr. Peter Heger, who ever since coming to Cedars has really invigorated the transplant biology study. And this is a heterotopic heart transplantation model. Basically, you take a mouse of one strain, in this case C57 Black 6, and then you transplant the heart from a biopsy mouse in the abdominal aorta. So this animal essentially has two hearts. And the whole idea is to see how long this second heart is able to survive, I believe by using contact palpitation, but I could be wrong. I don't think echo was used here. And of course, to prolong the model and give the heart a chance to survive, they also inject CTLA-4 Ig, which is a co-stimulation inhibitor, to allow the therapeutic to work, any therapeutic involved to work. And so the first very interesting observation we made was that animals that have specific deletion of TREX1 in myeloid cells have a much shorter graft survival compared to wild type. So this was already an indication that the TREX1 C-gas signaling pathway plays a prominent role in graft survival. Now, on the flip side, when you give TY1 to wild type animals, you find that the probability of graft survival increases quite significantly. We're not sure how yet, but this is still an active area of investigation. When Tiffany sort of did the dissection of the immune response, she finds that, as expected, the Ly6 C low macrophages, which are the less inflammatory macrophages, are much higher in the hearts of these animals, in the transplanted hearts of these animals, and the Ly6 C high is lower. Furthermore, she's also found that the CD8 positive T cells, or the cytotoxic T cells, which you know about far more than I do, are reduced in the hearts of animals that receive TY1, and the T regulatory, the FOXP3 positive T cells, the ones that are supposed to promote tolerance and graft survival, are increased. So these are all very encouraging readouts. And as I mentioned before, the mechanism has yet to be fully understood. But the conclusion of the work that I want to share with you today, very broadly, is that novel small RNAs and therapeutic extracellular vesicles can really inspire both emergent mechanistic understanding to the biology of the RNA itself, but also to the nature of the injury response and what pathways are involved and implicated, but also inspire the generation of new drug candidates. So this RNA that we're working with has a previously unknown biology and doesn't really fit in any of the general categories of an antisense oligo or an aptamer or an siRNA or any of the drugs that we see, RNA-based drugs that we see out there today. And that's cool because that's fairly disruptive. If we can alter the biology of the injured microenvironment in a novel way, that might give us some headway in places where translational efforts have met an impasse. So in future studies, we'll delve into the mechanism, what the crosstalk is. Of course, macrophage EVs are likely involved, but how much of that is influential versus direct contact through the immune synapse, time will tell. And then more broadly, the EV compartment is such a critical medium to understand the crosstalk between cells in any environment. And that could be the macrophage EVs signaling to T cells. It could be the T cell signaling to other myeloid cells. It could be the graft itself communicating with the host immune system. But, be that as it may, the RNAs that are there tell a story that yearn to be understood. And with that, I'd really like to thank all of you for your attention. I'd like to thank Peter for inviting me to this exciting event, if even virtually. I'd like to thank Tiffany for her wonderful work in the transplant data that I showed you. I'd like to thank, obviously, all members of my lab, the director of my institute, the support that we get from admin, and also our funding sources, California Stem Cell Agency, California Institute for Regenerative Medicine, the NHLBI, and I guess now what they call the Department of War, but once upon a time it was a DOD. And I look forward to your questions. Thank you.